Hi guys, welcome back to the video. We're just going to do a deep dive here, okay? So we're going to go through a few certain things of CRISPR-Cas9 in real detail. All right, so firstly the dot point, the function of CRISPR-Cas9 in bacteria and the application of this function in editing an organism's genome. So really this in-depth knowledge is probably not entirely required, but we're going to go through it. All right, again, CRISPR is your clustered regularly interspaced short palindrome repeats. We're going to skip this. All right, what is CRISPR? The important part is it's the adaptive immune response for a bacterium such as Escherichia coli. Okay, and what it does is it protects it from these bacteriophage viruses as well as any other viruses that are trying to infect it with their viral DNA. All right, and it uses restriction endonucleases. All right, so this is where we're going to go deep. So again, we're going to look at this image here. We have our bacteriophage here injecting its viral DNA. All right, and we have Cas1 and Cas2. So CRISPR-Cas1, CRISPR-Cas2. All right, and it's working to create that protospacer on this new piece of DNA. If this is an old piece of DNA, it would probably be a pink one. All right, this particular gene, or a blue one, this particular gene. All right, so Cas1 and Cas2 will find a particular spot. They'll find that PAM site, so the nucleotide guanine-guanine, so the protospacer adjacent motif. All right, that PAM site there, and they'll cut that out and they'll put that into the CRISPR array, which is really the bacteria's genome. Okay, you can see that this genome, well, in this case, this particular gene, has the promoter region where RNA polymerase will attach, has these repeats, and then it has these specific spaces. All right, and these are colored for a reason, right? Because they're all different for different viruses. All right, so that's the important part here. Really, it's just a immune system response. Alrighty. So we've looked at this here. Cas1 and Cas2 do their job if that's new. If that's not new, then there's a different process that occurs. Alrighty. And that process is really the creation of this guide RNA shown up here. All right. And that guide RNA fits perfectly into the Cas9 protein, which is going to be made upstream in the gene. Okay. Alrighty, so if we look at the bottom here, we have our CRISPR array. We have our promoter, repeat, the yellow spacer, repeat, pink spacer, repeat, blue spacer, repeat, etc., etc. Okay. We know RNA polymerase loves to bind on promoter regions, so that's what it does, and it will run down the strand. All right. Remember, this is a prokaryote, just like the trypopteron, so it's going to run down the strand, RNA polymerase, all right, and simultaneously at the same time, instead of a ribosome this time, because we're not making a protein, this extra enzyme RNase will then also run simultaneously. Okay, they occur together. All right, RNase is just after RNA polymerase. As the RNA polymerase transcribes this piece of DNA here in the CRISPR array, we call that the pre-CRISPR RNA. Okay, just like pre-messenger RNA, it's the same process there. All right, and as you can see, where the repeats are, they have these squiggly lines. Okay, these squiggly lines with, oh, what are they? They look very familiar, those loops. All right, there's hairpin loops. Very interesting. Okay, but in this case, it has nothing to do with the pulling on the strand, just like trypopteron. It's a little bit different. All right, so we have our pre-CRISPR RNA, as you can see here. All right. Alrighty, and from there, that needs to be transcribed by this RNase enzyme. All right, that will run down the strand. Okay, it'll run down the strand here. All right, and it will transcribe that spacer RNA, as well as it will transcribe a little bit of the spacer itself, and then a little bit of these hairpin loops. Okay, so we call that the spacer RNA. So as you can see, the specific segment, and then we have the tracer RNA. Okay, and they will come together to make CRISPR tracer RNA. So firstly, it becomes the CRISPR RNA here, and then it attaches with the tracer, which makes a CRISPR tracer RNA. Okay, and again, CRISPR tracer RNA, really, that is the same thing as your guide RNA. Okay, as you can see, this is exactly the same as this. All right, and then what will that do? That will do its job. All right, CRISPR tracer RNA, also known as the gRNA, will then lock into, as you can see with this little hook-shaped loop part. All right, that hooks into the Cas9 enzyme. All right, and really that just hooks in, and then the gRNA, sorry, the spacer RNA part, which is the yellow part that we looked at. All right, all the way from here to here to here to here. All right, that spacer RNA part, that is the specific sequence. All right, Cas9 will come along to a specific complementary sequence to this gRNA, and it will cut a couple bases down. So normally it's 3 to 5 bases down from the PAM site. Okay. So again, this is showing the CRISPR-Cas9 enzyme. All right. So you have the Cas9 behind here. Cas9. Alrighty. And then of course you have your gRNA. Or guide RNA. Alright. And that's made from the CRISPR tracer RNA. Okay, as you can see, it hooks in here to this part of the polypeptide, so this orange polypeptide. Obviously, this Cas9 enzyme is a quaternary structure polypeptide. All right, this is our complementary part here, which was our original spacer RNA, which was saved by Cas1 and Cas2. All right, and that will cut, so snip, snip, a few bases down from the PAM site that it finds. Alrighty. So really, that's the deep dive. All right. That's as deep as we really need to know it. Again, CRISPR-Cas9 can be harnessed by scientists by combining it with our own human-made gRNA. Okay. Guide RNA is a short length of code that unzips the DNA. As you can see, this is the process of the Cas9 enzyme with, obviously, it's CRISPR. It's CRISPR guide RNA. That's the process of it cutting. All right. So guide RNA is a short length of code that unzips the DNA. So as you can see, the DNA is unzipping here. And it's just searching. It's searching for whether these are complementary. All right, so it'll go around testing pieces of DNA. Obviously, the bacteria's own genome won't have that particular complementary bases, as well as the PAM site. And obviously, the viral DNA will have this. Okay, the viral DNA has it. It finds that PAM site, and then it goes a couple of bases down, snip, snip. All right, bye-bye to that piece of DNA. Once it's snipped there, it is no longer working. All right, it's unexpressible from there. Okay, therefore, no more viruses are being made in the cell because that viral DNA is not doing its job. Okay, that was the deep dive video. Hopefully that cleared up a few things for you. I didn't go through everything in the last video. I wanted to make it a bit more simplistic, but it seems like everyone's watching that one, so I just wanted to do this too. Is there anything else I need to show? No, this is the usual stuff. This is the same stuff we looked at last class. Okay. Obviously, scientists can use this to make GMOs. That's pretty important. Okay. Awesome stuff. If you haven't done these things, do it. If not, have a nice day. Take care.